Okay, so ladies and gentlemen, you are all welcome, and I think we are set and we can start the tutorial or the revision. So, and I hope it will be of beneficial to all of us. So, ladies and gentlemen, first, I think what I am going to do for the today's class, I'm going to answer some basic questions for the course, and I'm going to have another like 3 hours to summarize the whole course from beginning to the end. So I'm going to use like 3 hours to summarize all the course from beginning to the end. So as a result of that, I will advise you uh, to read the course very well, because it is, number one, it is a very complicated course, and in addition, it also very bulky. So as a result of that, we are actually going to look at the course very well and do justice to it. So I think one of you has actually sent some questions to me in order to actually look at them. So without wasting much of my time, I think I will actually look at the question. I will look at the questions and then we summarize some of the questions. So I think, let me start reading the question. Number one, the first question says, enumerate any three examples, or let me just read the title of the questions. So the title of the course is Biochem 241, and it is, of course, the year for the exams is 2024 May, 2024 May, weekend group. So the first question says, enumerate any three examples of physiologically most important: one, monosaccharides; two, disaccharides. So the first question says, enumerate the two important, physiologically important, one, monosaccharide and then polysaccharides. So ladies and gentlemen, the first one, when we said polysaccharides, or when we hear the word saccharide, it means sugar. And this sugar, we have what you call, or generally, sugar is a carbohydrate. So what is carbohydrate? When you have the word carbohydrate, the best way to describe carbohydrate, there are polyhydroxyaldehydes or ketones, meaning that for a compound to be qualified or to call a sugar or a carbohydrate, it must have aldehyde or ketone functional group with many OH group. And we have three different classes of carbohydrates. We have we have monosaccharide, and then we have oligosaccharide, and then we have polysaccharide. So monosaccharide are the most simplest sugar, and the most important, physiologically important monosaccharide that is asked in this question, we have number one. The first one is glucose, and then the second one is fructose, and then the third one is galactose. So these are the three most important carbohydrate or monosaccharide that are of physiologically important. So that is for the first question. Under question one, that is the answer to the Roman figure 1. So the important physiologically monosaccharides, we have glucose, we have fructose, and we have galactose. Why are they called physiologically important monosaccharides? Because they are monosaccharides that are utilized by the body in terms of energy generation, in terms of the maintenance of the blood glucose level. So these are the important physiologically monosaccharides. And then two, they said the physiologically important disaccharides. So what are the important physiologically important disaccharides? Disaccharides, they are sugars that have two simple sugars. So when you have two simple sugars, when you have two simple sugars, that is two monosaccharides joined together, they will form or they will generate the disaccharide. So when you said disaccharides, disaccharides are formed as a result of combination of two monosaccharides. And how these two monosaccharides form? They are formed as a result of glycosidic bonds. So don't forget about this. So disaccharides are formed as a result of combination of two monosaccharides through glycosidic bond. So this disaccharide is under oligosaccharide. So remember I told you that we have three types of sugars or three types of carbohydrate. Number one, I said we have monosaccharides, and number two, I said we have oligosaccharide, and then the last one is polysaccharide. So when we said oligosaccharide, there are sugars that contain 2 to 15 monosaccharide. So when you have 2 to 15 monosaccharide, that types of sugars, they are called oligosaccharide. So therefore, the disaccharide itself is under oligosaccharide, because we said oligosaccharide, it contains 2 to 15 sugars. So since disaccharide contains only 2 sugars, it means that it falls under oligosaccharide. So what is the answer to this question? So the two importance or the three importance the two physiologically important oligosaccharides, we have number one, disaccharide, we have maltose, we have lactose, and then we have sucrose. So these are the three important disaccharides that are found in living organisms. So number one, I said we have lactose, we have sucrose, and glucose. So fructose and glucose is what actually form the sucrose. So the fructose and glucose, so you should get that. It's very important. So for each of the disaccharide, you have to know the constituent monosaccharides that form them. And then what about maltose? Maltose is made of by two molecules of glucose. So two molecules of glucose linked together by glycosidic bond is what form the maltose. And then the lactose is a combination of glucose and galactose. So these are the two disaccharides that are of physiological importance. So you see now we answer question 1, Roman figure 2. Then now B, name one hexose sugar that originated from fruits. So which of the sugars that is formed or that is originated from fruit, the hexose sugar? Please, who can answer this question? Let someone answer it on the comment section so that we will be answering the question together. The hexose sugar, that is the 6 carbon sugars that is actually generated from fruit. Please, who can answer the question? I'm seeing, please, someone should answer it from the comment section. Excellent, excellent, excellent. So the answer is fructose. So I think that is what I will be doing. I'll be asking you the question and then you will be responding. So thank you very much. The answer to this is fructose. So let me also read another question. Differentiate between prokaryotic cell and eukaryotic cells. So who can differentiate between prokaryotic cell and eukaryotic cells? The difference between prokaryotic cell and eukaryotic cell. Yes? The difference between eukaryotic cell and prokaryotic cell is the next question. Yes, Achiba Jasmine, thank you very much. Evening. Please, if you didn't join or if you didn't join the YouTube channel, please try to join. Okay, so Mina Solomon Eli said prokaryotic cells are not membrane bound. Mina, this is not correct. Prokaryotic don't have nucleus, while eukaryotic have nucleus, according to Ilya. So this is partial. I can say is partial, your answer. Then Kena said prokaryotic cell has no nucleus, while eukaryotic has nucleus. This one is also partial. Then eukaryotic cell has nucleus, while prokaryotic lack nucleus. This one is also partial. So eukaryotic cell have nucleus, which is covered by nuclear membrane, while prokaryotic cell have typical nucleus. So I think, let me just answer this question so that we will move to the next. Prokaryotic cells are cells that doesn't have nucleus and membrane bound organelles. Prokaryotic cells are cells that doesn't have nucleus and membrane bound organelles. So you forget with the membrane bound organelles. So what are the membrane bound organelles? Membrane bound organelles include Golgi body, mitochondria, lysosomes, ribosomes. They are all under uh, membrane-bound organelles. So any organism that doesn't have nucleus and membrane-bound organelles, that organism is called prokaryotic cell. An example of prokaryotic cells is bacteria. Bacteria and archaea bacteria, they are all under prokaryotic cell. And then for the eukaryotic cells, there are cells that have nucleus and membrane-bound organelles like our own cell. So this is the answer to that. This is the answer to that question. So, ladies and gentlemen, if this is the first time you are coming to my YouTube channel, try to join the YouTube community. Try to join the YouTube community, as I'm going to do a video. I'm going to summarize the course, the whole course, in 3 hours from beginning to the end. I'm going to use a bot summarizing everything. So if you didn't join the YouTube channel, please try to join so that at least you will have access to all my videos for this semester and the second semester. To try to join. So then, so we are done with question one. Seriously, this question paper is very, very simple. No, Achiever, just me and you already joined. I can see it from here. You are already the member to my YouTube channel, so you already joined. So it's only for those that have not joined, then let them join. I'm going to summarize the video, the whole course in 3 hours, then I will upload it. So only those that joined can only see the videos. So then we are moving to question 2. Name 5 cell organelles and describe the functions of any 3 of them. Yes, expensive Diago, I'm going to summarize the whole course in 3 hours. Yes, I'm going to do that by God's grace. And I will do that possibly tomorrow or Monday. So the next question, which is under question 2, says name 5 cells organelles and describe the functions of any 3 of them. Okay. Nancy says mitochondria power. So first you mention them. So after you mention them, then their function. So I'm seeing what are the 5 membrane bound, or sorry, what are the 5 cell organelles. So let someone mention them. The 5 cellular organelles. Yes, mitochondria. Mitochondria is one of the organelles of the cells. Then which one again? You have been mentioned. Okay, lysosomes, mitochondria, Golgi body. Let someone mention them all at once, man. You should mention them all at once. Mention them at once. No, Kolasade Adebola, you didn't join. Mention all the 5 organelles. Okay, excellent, Diago. You said we have mitochondria, Golgi body, nucleus, rough endoplasmic reticulum. So excellent. So now, outside that 5, we are to explain 3. We are to explain three. So let's start with mitochondria. What is the functions of mitochondria in a cell? The functions of mitochondria in a cell. Yes, the functions of mitochondria. Waiting, I'm waiting. Excellent. Achiever is the powerhouse of the cell. So mitochondria is the powerhouse of the cell. Why is it that mitochondria is considered as a powerhouse of the cell? Because it is the site of the cells where most of the energy are generated in the form of triphosphate. So adenosine triphosphate is the energy that we use to survive, the energy that we use to do every activities that we are doing. So that is why mitochondria is considered as a powerhouse of the cell. And in addition, apart from mitochondria is considered as a powerhouse of the cell, it is also considered as what? As the organelles that also contain genetic material. So we have three organelles of the cells where the genetic materials are located. These three genetics material these organelles that contain three genetics material include nucleus, mitochondria, and chloroplast. So it means that the mitochondria have two major functions. One, it contains a circular genetic material, it contains DNA, and it's also considered as a powerhouse of the cell. It generates most of the energy that we are using. But apart from the mitochondria, where do we also generate energy? Cytoplasm also. In the cytoplasm, we also generate energy. Yes, adenike, adelao. Yes, nucleus, mitochondria, and chloroplast. That is where the genetics materials reside in a cell. So after this, then the next thing is we have mentioned the five organelles and we have talked about the functions of the mitochondria. So what is the functions of ribosomes? So excellent, Tylo. You said ribosomes makes protein synthesis. No, the English is not correct. Why can't you just say protein synthesis? So the ribosomes is the protein industry of the cell. So the proteins that are actually synthesized in our body or that is synthesized in our cell is the ribosome. Then which other organelles? So you have explained the functions of two organelles. So which one again? Which other cellular organelles that you can mention or that you can explain the function apart from we have talked about mitochondria, we have talked about okay, Philip said, how can I join? What you are going to do if you check the YouTube channel, you are going to see a place where it wrote join. So to join, you have to use your ATM card. It's just 750 naira. Okay. Isaac Elizabeth says the nucleus direct all the activities of the cell. So you are correct. So this is no Beatrice Dobe, you didn't join. You didn't join. So excellent. You see, Ade Solar just joined. So you are welcome. You are now a member. So if you now join this YouTube community as a member, you are going to have access to your second semester biochemistry course. That is the general metabolism. You are going to have access to our physiology courses as well as the general microbiology. So you are going to have access to them. So, uh, so I think we are done with that. So let's move to 2B. Explain the functions of plasma membrane. Explain the functions of plasma membrane. Yes, of course, Ogo, you are going to get the video the link to this video revision. Of course, you are going to get it. So now the question is explain the functions of plasma membrane. So who can tell me who can list three important functions of plasma membrane? That is the cell membrane. When we said plasma membrane, it means cell membrane. Excellent. Diago said Diago, you are actually a very good student. He said plasma membrane is for protection and transportation of materials in a cell. And also, uh, a tank is said it regulates what goes in and out of the cell. Yes, you are right. So a plasma membrane, it first protect the cell itself. Because if you look at the cell, it is covered by membrane, by a cell membrane. So the cell membrane, it covers the entire cell. So as a result of that, it protects the cell from any external attack. And there are materials that some we need them inside our cell, and some materials we doesn't or we don't need them in our cell. So the cell membrane, it is the function of the cell membrane to control what will get into the cell and what will go out of the cell. So it is excellent. So that is the selective permeability. So when we said selective permeability, it means to select what will get into the cell and what will go out of the cell. So that is the two functions of the cell membrane that we mentioned. One, it protects the cell, and second, it select what will get into the cell and what will go out of the cell. So these are the two functions of the cell membrane. And then third, cellular recognition. Excellent. So when it says cellular recognition, what is cellular recognition? There is a way that each and every cells of the body interact with one another, communicate with one another. So the cell membrane helps in recognizing one cell, and it also helps in communicating with another cell. Like for example, let's say you have these cells and this cells is infected with virus, and you have another cells like this which is not infected. So it is the membrane of these infected cells that will send a signal, it will send a message to these cells through the membrane of these cells to help them communicate, to help them communicate that, okay, I'm infected with a virus. So I'm communicating with you so that to try to develop a mechanisms that will protect you against anything that will affect you. So that is the role of the cell membrane. So it helps in cellular recognition. So apart from the protection, transportation of the material, and then communications, there is also cellular recognition. So communication and cellular recognition are also part of the functions of the cell membrane. Excellent. So Husseina Samaila said it also protect the cell integrity. That is the protection. So, ladies and gentlemen, I think we have made mention like 5 different functions of the cell membrane. Yes, it also gives the shape to the cell. Okay, Omolade is asking on how to join the YouTube community. So, what you are going to do now, if you check the YouTube, you will see join. So, on that, you click on the joint, you use your ATM card, you use those digits at the back. So you put them there and then you click. So 750 naira will be deducted from your account. Yes, look at Basilia just joined. Basilia EK Basilia just joined. So the importance of joining the YouTube community, we have a lot of videos. In fact, even now, if you join the YouTube community, there are some videos that I use or that I solve some questions. So you can have access to them. So once you join, you will have access to some of those videos. And I'm still uploading, and I will even upload another one. So can you give us the grace till after exam? I don't understand. What do you mean the grace till after exam? Please explain. So list 3 contributions of biochemistry to to the nursing science. So, what are the three contributions of biochemistry to the nursing science? Please, who can mention the three contributions of biochemistry in nursing science? Yes, answer these questions on the comment section. Um, one day, Betty, join the YouTube community is not compulsory. Is for your interest. So for all those that are interested and they are willing to join, it's optional. It's optional. Yeah. So I'm not saying that it's compulsory. So it's for those that are actually wanted to join. The importance of the YouTube community or the YouTube channel, it's in the YouTube I actually teach biology, I teach biochemistry, I teach physiology, and I teach anatomy. So there are all those videos in my YouTube channel. So it's for you to have access, to always have access to our videos, then you join. And it's not only for the LC student, it's for global. There are some people that joined from I think after this tutorial. Okay, there is also one member that just joined. Edna, you are welcome for joining the YouTube community. So what I wanted to is for global interest. Some people from UK, some people from India, some people from different part of the world are actually watching my videos, and they are members of my YouTube community. So I'm actually uploading my videos for the for the international interest. So it's not compulsory. Can you incorporate other lecturers to summarize other courses, other other courses? Where is it? I saw one. Why no, I can't tell other lecturers to summarize their own courses. What I know is, I know what I only do what I can do. So I can't convince someone to do what he don't wanted to do. So actually, it's my it's my wish, and this is my YouTube channel, and I opened this YouTube channel I think since 2016, but I actually make it very active last year. So I can't convince anyone to do what I'm doing. It is my wish, and I don't know what others will do. So I think some of you have answered this question. The question says, list three contributions of biochemistry to nursing sciences. So you know that in nursing, you are involved in disease diagnosis, in disease treatment, as well as drug development. So actually, the major role of biochemistry in nursing science, number one, it helps in disease diagnosis. It helps you to diagnose disease. It also helps you to treat disease. In fact, with the knowledge of biochemistry, you can even develop drugs. Excellent. Someone said nutritional management. Yes, of course. With the knowledge of biochemistry, you can actually treat diseases. You can treat disease. You can treat disease. Okay, Adewale, you are welcome. Another person has just joined. So I'm just actually advising you to join so that at least you know that immediately after these exams, you are going to join another semester. So all videos for your next semester will be dropped there. They are there. So if you are a member, you can expensive. So it helps in nutritional management. What is nutritional management? When you say nutritional management, it means how you can use nutrients or food to treat diseases. Like for example, we know that when it comes to the issue of let's actually look at the nutrition, like sexual dysfunction. Sexual dysfunctions, there are a lot of people that are suffering from sexual dysfunction. So, with the knowledge of nutrition and biochemistry, you can be able to actually put your patient on you can put your patient on particular diet, and it can definitely cure their disease. Like for example, ulcer, if you can use alkaline food, because most of the ulcer, it caused uh, as a result of increase in hydrochloric acid, or in the or as a result of the activation of pepsinogen by hydrochloric acid. So if you can get a alkaline foods like apple, it can definitely reduce the crisis of ulcer. In the case of sexual dysfunction, if you have someone that actually have a problem of sexual dysfunction, maybe he cannot actually satisfy his family, or maybe she cannot satisfy her husband, then of course there are class of nutrients that you can use to treat disease, like chocolates, like granules, and so many others. How can I subscribe to the channel, sir? Yes, what you are going to do, you will see join. So you click on the join, then you use your ATM card. There are 15 those numbers at the back of your ATM. Then you use them. You will It's just only 750 naira that will be deducted. So that is that. So the next question, I think you have answered this: disease treatment, nutritional management, disease diagnosis. All are the actually contributions of biochemistry in nursing. So then, so we are done with question two. We are done with question two. So I'm moving to question three. Okay, Dennis, you are welcome. Thank you for joining. So, highlight the relevance of liposomes as a novel drug delivery tools. So, what is liposomes? Liposomes, the lipo actually originated from lipids. So, you know that uh, most of the lipid particles, usually they are insoluble in water. Remember from the basic definition of lipid, what is lipid? A lipid is a molecules that is insoluble in water but soluble in organic solvents. Okay, Olive Funke, you are welcome. Thank you for joining. So it is insoluble in water but soluble in organic solvents. So they are insoluble in water but soluble in organic solvent. So but if you look our body fluids generally, our body fluids is water, or is hydrophilic. So any lipid substances, it cannot dissolve, uh, it cannot interact with the lipids or lipid particles that we are taking. So that is why, like drugs, most of the drugs, they are lipophilic in nature. Most of our drugs, they are lipophilic in nature. When we say lipophilic, it means that they are lipid soluble. So how some of these drugs that are lipid solubles get into our body is by actually conjugate them with the lipids, making them to form what you call a complex called liposomes, so that the drugs can easily be delivered into our body. So that is the importance of liposomes. And then the next thing, describe the techniques involved in the purification of proteins. So protein purification. What is protein purification? So generally, ladies and gentlemen, it is very, very important to understand or to get the concept of protein purification. Generally, most of the proteins that we have, or most of the proteins that are found in living organisms, they are reside in cellular organelles. Gloria Oluka, you are welcome to the YouTube community. So what I'm saying, ladies and gentlemen, please get this concept is very important. It depends on the, like for example, okay, I think let me just give this example is the simplest way. You know that we have diabetes type 1. So what happened to diabetes type 1? Diabetes type 1 is a disease that affects teenagers and is usually occur as a result of insufficient insulin secretion. Insufficient insulin secretion. So now assuming now you have a patient and you, before, because now we have a technology, we have what you call recombinant DNA technology and molecular biology that enables scientists to generate insulin in an industrial scale in industry. So using the knowledge of molecular biology and biotechnology, you can produce insulin industrially. That is why you don't need to suffer yourself into this step that I'm going to explain, but I'm going to explain it now. So assuming now you have someone that is suffering from, uh, let's say you have someone that is suffering from diabetes, so you need insulin. So assuming now you discovered a bacteria that contain or that produce insulin. So what are you going to do? So the first instance, you know that okay, this is a bacteria that can produce insulin. Get the isolate of that bacteria, cultivate them in the laboratory so that you have the load of that bacteria in high concentration. So what are you going to do? Try to understand where the bacteria produce, or the exact location where the bacteria produce that uh, insulin. So what you are going to do, you open up the bacteria, and you should know that in the bacteria, it is not only that insulin. There are so many other proteins. So when we said protein purification or protein isolation, it means the techniques that you are going to apply to get your protein of interest. To get your protein of interest, that is protein purification. Let's say there are 1,000 proteins in the bacterial cell. So the protein purification is aimed to isolate only your protein of interest. Let's say now the protein is in human and is in the pancreas, like in the case of insulin in human is in the pancreas. So how are you going to isolate insulin from human pancreas? So the first thing you need to kill the human, of course, or you need to harvest the pancreas. So after you harvest the pancreas, what do you do next? Then you open up the pancreas. So in the pancreas, you know that we have beta cells, we have alpha cell, we have epsilon cells. So what are you going to do? So you have to open up the pancreas. After you open up the pancreas, then what do you do next? You isolate the beta cells because pancreas are produced in the beta cells. Okoro onye kachi, Rachel Obi, Eleven Gang, you are all welcome to my YouTube channel. So after you isolate the beta cells from the pancreas, what next? Then you get an isolation medium where you are going to open up the beta cells. So when you open up the beta cells, all the protein content of the beta cells will be released into the isolation media. So if it once it released into the isolation media, what will happen? There are so many proteins and also other non-protein components. So now I'm going to start explaining the techniques. So now You get the pancreas, you open up the pancreas, you isolate the proteins. So after you do that, it means that there are protein component and there are non-protein component. So what next? Then the best techniques that you are going to apply, ladies and gentlemen, is because in the beta cells, after you open up the beta cells is a eukaryotic cell. It means that there is a mitochondria, there is a nucleus there is Golgi body, there are ribosomes, there are lysosomes, there are so many of them. So the best techniques that you are going to apply after opening up the beta cells, you apply centrifugation, you centrifuge. So after you centrifuge, you spin, you are going to have different component of cells. You are going to have nucleus, you are going to have mitochondria, you are going to have lysosomes, you are going to have your ribosome. So assuming your protein of interest is in the cytoplasm. So if your protein of interest is in the cytoplasm, what are you going to do? So after centrifuging, all other components are heavier than the cytoplasm, so they will be settled down. So after they settle down, what are you going to do? It means that the supernatant is your cytoplasm. So you are going to take out your cytoplasm. So in the cytoplasm, you have your insulin or you have your protein of interest and other non-protein component with also other proteins. So what do you do? Then you apply. So you see, first you start with centrifugation. So after centrifugation is ammonium precipitation. Ammonium precipitation. So what is ammonium precipitation? So you see, I said you have your cytoplasm which contain proteins and non-protein components. So the importance of adding ammonium phosphate is the salt that is used in ammonium precipitation. So once you add ammonium phosphate, all the proteins will precipitate, they will clump together, and the non-protein component will be separated. So you see, you have protein component and you have non-protein component. So what you are after is the protein component because your protein of interest is part of those protein component. Then you discard the non-protein component. So after you discard the non-protein component, what next is dialysis. So what is the importance of dialysis? The importance of dialysis, you will take your protein and put it into a dialysis bag. And in the dialysis bag, there are small pores. So you immerse your dialysis bag into a buffer. So what is the importance of the dialysis? You see, you use ammonium precipitate. So those ammonium precipitate, there are salts and there are small molecules, and you don't need them. So the importance of that dialysis is to remove those salts, so that you will only left with the protein. Okay, Muhammad Al Mustafa, please sir, I don't know how to join. So actually what we are going to do to join, you you will see joint. So you click on the joint to use your ATM card and 750 naira will be deducted. It's not compulsory, it's for your own interest if you want to join the YouTube community. And the importance of joining the YouTube, it will actually assist you in your physiology courses, general biochemistry that you are going to do next semester. So if you join this YouTube community, very soon, maybe in the next 2 weeks or 3 weeks after your exam, then you will start seeing videos on your second semester courses that you are going to do. So if you are a member, you will always get notification. You will always get notification. So that is why I'm just advising you, it's just 750 naira. You'll see join, so you click on the join, then you use those number at the back of your ATM, then you can easily join. So you see, okay, Helen Ebo, you are welcome. So someone just joined. So now, ladies and gentlemen, we are now done with dialysis. So you remove the protein component, you remove the non-protein component from the protein. You remove the small molecules like the ammonium salt. So you are now left with protein. So what next? So there are also other techniques like affinity chromatography. I'm going to explain this briefly. And if you need if you need this video on protein purification, I have it. I have 1 hour video on protein purification. So you can actually, if you want the videos, then I can send it to you. So if you want it, I can send it to you in the group. So you have affinity chromatography. The importance of affinity chromatography is to remove or is to isolate proteins based on their binding. The principle is based on their binding. So let's say you have a thousands of proteins. So what we are going to do in the affinity chromatography, you are going to add a ligand in the column. So the ligand will be in the column, and then your protein of interest will only specifically bind to that ligand, and the other non-protein or the other protein that you are not interested will be eluted. And ladies and gentlemen, sometimes affinity chromatography is the best, provided that you have the knowledge, you know your protein of interest. From there, you can easily get the ligand that can specifically bind to your protein of interest. So that is for the affinity chromatography. And there is also ion exchange chromatography. The importance of ion exchange chromatography is to separate proteins based on their charges. I'm just explaining this briefly. I have the full video. If you want, then I can send you the link. And there is also column chromatography based on the mobility of the proteins. So after you do that, then at the end, you are going to separate your proteins and you are going to have your protein of interest. So if you want, the next thing you can even, if you don't have an idea of the amino acid sequence, then you can sequence your protein. So how do you sequence your proteins? You can sequence proteins using Edman reagents or using Sanger methods. And there's a conventional way of doing that. There are some chemicals like trypsin, like carboxy 50 days enzymes. There are so many of them. I'm not saying that this lecture is not for the next semester. This lecture is for this semester Biochem 241. Biochem 241. What I'm saying, if you now join as a member to this YouTube, you are going to have access with even your second semester videos. So this is Biochem 241. So I also have a video on amino acid sequence. And I know that I saw one question that they asked a questions like this. Yes, of course you can join tomorrow. Anytime you want to join, you can join. So that is actually for the affinity chromatodes. That is actually for the protein purification. So you can even, so ladies and gentlemen, I think I will just advise you this. I will send you the video for amino acid sequencing because I saw a year where they asked a questions like this, and it's also in your courseware. It is also in your courseware. But I think I have like a video of 26 minutes that will explain to you on how to do the how to sequence protein. So I think it's important. You can just glimpse through and understand, because there was a year that they asked which of the proteins, which one are acting on the N terminal and the C terminal. Yes, Beatrice Dobe, you have joined. So thank you. This is Chuku Olubube. You have also joined. So you are welcome to the YouTube Alheri Sofa. You are also welcome to the YouTube community. So that is that for the affinity chromatography. So you see that the question says describe the techniques involved in purification of proteins. So I mentioned the techniques. One, centrifugation is the techniques. Second, affinity chromatography, column chromatography, ammonium precipitation is also amino acid sequences. All these are the techniques that are involved in protein purification. So don't forget about them. So I mentioned them: centrifugation, precipitation, affinity chromatography, ion exchange chromatography, column chromatography. All these are are the techniques that are involved in protein purification. So now we are moving to question 3C. So a protein can be sequenced using which process? Okay, you can use Sanger sequencing method. You can also use Edman sequencing method. So these are the two most used methods of protein sequencing. Okay. So now the next one is 3C. Although the question here is not clear, but it's like it said that states the main purpose of protein purification. State the main purpose of protein purification. Please, my friends, please, who can tell me what is the main purpose of protein purification? I said it. So please, who can tell me the main purpose of protein purification? My monitor Abakar, you are welcome. So you just joined the YouTube community, so you are welcome. Okay, Mina Solomon Eli, you can ask your question. But as you are asking your question, I want someone to answer this question because I have explained it while explaining the protein purification. State the main purpose of protein purification. Please, who can state the main purpose of protein purification? Yes, Diago, you are always doing excellent and good. The main aim is to only get the protein of interest in a pure form, to get the protein of interest in relatively pure form. That is the aim of protein purification. The main purpose is to increase the protein specific activity to the value expected for the pure protein. Yeah, this one is also very interesting. Yes, specific activity. What is specific activity? Yeah, I have lecture on enzymology. That is where you are going to know the specific activity and enzymes activity. So it's under your last topic, enzymology. So I have a good full video on that too. So I think that is the answer. That is the answer. So this look at I this is there are question 4A. The questions here are very, very simple. Let me just read them so that I will get the answer from you. The question says, state and explain the type of bond associated with the following macromolecules. State and explain the type of bond associated with the following macromolecules. A. Nucleic acids. Who can tell me what is the types of bond that is actually associated with nucleic acids? Yeah, as I said, for those of you that wanted to join, I said you should just check on on the YouTube channel join. So when you see join, then you use your 8 videos on my YouTube page. So someone said glycosidic bond. Martins Abigail is not glycosidic bonds. Nucleic acids. Okay, Dorcas, as a you are welcome. So another person have just joined. Okay, Mina. Okay, your question: the techniques of protein purification, is it all the stages we are to write, or we pick one of the method and discuss it. They said, okay, let me read the question again. Describe the techniques, not one, the techniques involved in the protein purification or in the purification of proteins. So it's not one. So you mentioned as many, like five. Like even I mentioned like five. Yes. So please, who can answer this? The types of bonds associated with the following macromolecules. So what is the types of bonds that is found in nucleic acids that joins or that form the nucleic acid? So which types of bond is that? That is how one nucleotide joins with another nucleotide. No, Queen Osuji is not peptide bond. The nuclear acid bond are DNA and RNA. No. Excellent, excellent, excellent. Oni Orisan. The answer is phosphodiester bonds. Phosphodiester bond. So what actually form the nucleic acid? The building blocks of nucleic acid are nucleotides. So nucleotides are joined together through a phosphodiester bond. So please don't forget about this. Don't forget this. One nucleotide joined with another nucleotide through peptide bonds. Yeah. And how these peptide bonds are formed. The 3' of one sugar joined with a 5' or the 5' of one sugar and the 3' of another sugar joined together through phosphodiester bond. So the answer is phosphodiester bond. Then, what are the types of bonds that are found in carbohydrates? The types of bonds that are found in carbohydrates. Please, who can answer this? Excellent, Isaac Elizabeth. The answer is glycosidic bond. Excellent. No, it's not covalent bond, Stephanie. It's glycosidic bond. It's glycosidic bond. Excellent. So then what of proteins? What types of bond is formed in proteins? The types of bond in protein. Excellent, Kaulat Ahmad. The answer is peptide bonds. So the type of bond that is found in proteins is peptide bond. Yes, Ijafa. So for those of you that have problem with joining, what I said, just check on the YouTube. There is you'll see an icon, join. Then you click on it. Then you use your digit at the back of your ATM, and then you join. So we are done with question 4A. So this is question 4A. This is question 4A. So we are now moving to question 4B. Mention the major application of synthetic nucleotides. What are the major applications of synthetic nucleotides? Okay, for Lasade Adebola, you can try it again. Synthetic nucleotides. I'm waiting for an answer. Synthetic nucleotides. Okay, Diago, no answer. So who can assist? Okay, Juliet, you are welcome. Juliet just joined. It can be used to determine genetic disorder. Excellent. So briefly, you can say that synthetic nucleotide can be used in scientific research. So you can use it in scientific research. And you can also use it uh, in treatment of disease as well as the treatment of genetic disorders. So you see, number one, you can use synthetic nucleotide in medicine for the treatment of disease and genetic disorders. So you can use synthetic nucleotide to treat disease and genetic disorders. And some of these diseases that can be treated with synthetic nucleotide, you can treat HIV, you can treat hepatitis infection. In fact, even sickle cell anemia, you can use these synthetic nucleotides to do that. And then in addition, you can also use these synthetic nucleotides in agriculture for insect resistance or for pest resistance. So this is actually some of the applications of synthetic nucleotides. Yeah, I used to study DNA polymerase dynamics and specificity, and my even inhibit DNA polymerase activities. Yes, excellent. So these are some of the applications. You can use it in information technology. You can also use it in NASA. So all these are applications of synthetic nucleotide. So let me read the next question. Yes, in diagnostic application. Yes, Izank. So thank you very much. And to question 5A, or we should stop here. Can we continue? Or we should continue? Okay, so we should continue. So let's move to question 5. Give the name and the structure of one only non-chiral amino acid. So which of the amino acid it is non-chiral? You know that we have 20 amino acids that are found in protein. So almost all those amino acids have, and what is a chiral carbon? A chiral carbon is a carbon that have 4 different groups attached to it. So which of the amino acids doesn't have a chiral carbon? The only amino acid that doesn't have chiral carbon. Please answer. Don't forget to join the YouTube community. As I said, I'm going to summarize the whole course. I'm going to summarize the whole course in 3 hours. So if you didn't join the YouTube community, try to join so that at least when I upload the video, you can have access to it. What is the amino acid? Which of the amino acid that doesn't have chiral carbon? Excellent, Sahir Hamisu Madaki. I'm surprised, Sahir, you didn't join the YouTube. So the answer is glycine. So if you check the structure of glycine, you'll see that there is a chiral carbon, and this chiral carbon, sorry, there is a carbon, and the central carbon have 2 different hydrogen atoms attached to it. So as a result of that, the carbon become non-chiral. Because for the carbon to be a chiral carbon, it must have 4 different groups attached to it. Yes, Stephanie Moses, yeah, of course, we can use synthetic nucleotides in nanotechnology, that is in the information system. So the answer is glycine, then you are right. So let's look at another question. 5. I can actually, as AFMAS, glucose, galactose, and mannose show the structural differences between these molecules. So you see, when we said AFMAS, what are AFMAS? Compound have the same molecular formula. They have the same molecular formula, but structure of glucose, galactose, and mannose. You will see that they are all the same. The only His-Salvage equation. So what is Henderson His-Salvage equation? So I think for the Henderson His-Salvage equation, I also have a video. It's under uh, BOFA, acid, base, and BOFA. So I have a video. I think I will send you the video on that if you are interested. It is a formula which says pH is equal to pKa plus logarithm of conjugate base divided by the concentration of the acid. Let's say I have a question. Draw the structure of polypeptide containing alanine, glycine, alanine, glutamate, glycine, and proline. So this is actually one, two, three, four. This is actually a tetrapeptide where you are going to have alanine join with glutamate. Sorry, join with glutamine, join with glycine, and then proline. And at the end, the name is going to change, which is going to be alanyl, glutamyl, glyceryl, proline. So that will be the name. So state and define each of the terms. So I'm going to send you a video on that. And then the next thing is question 6, which is the last one. Outline the 6 types of cofactors required by most enzymes. So you see, enzymes generally are organic molecules that speed the rate of the reaction. They are molecules that speed the rate of the reaction, and some of these enzymes are usually in the form of proenzymes. That is, they are inactive. They are inactive, and for them to actually become active, they need cofactors. So what are the cofactors that are required by most of the enzymes? So the 3 types of cofactors that are required by the coenzymes, although the cofactors are divided into 2, we have 2 types of them. We have prosthetic group and we have coenzymes. So we have nicotine, we have even coenzymes E. So these are the examples of the cofactors that are required by the enzymes for them to work maximally. Then enumerate the 3 isozymes with wide clinical applications. The 3 isozymes that we have that are used for wide medical application or clinical application. So when you said isozymes, they are enzymes. They are enzymes that catalyze the same types of reaction, but they are located or they are in different part of the body. Like for example, if you take lactate dehydrogenase, we have it in the heart, we have it in the liver, we have it in the muscles, but they are all in different locations. But they are all catalyzing the same type of reaction. So when you said isozymes are enzymes that catalyze the same type of reaction, but they are at different part of the body. They are located differently. So with the lactate dehydrogenase, you can use it for the diagnosis of myocardial infarction. So this is the examples of the isozymes that can be used or that have clinical application. So that is question 6b. So for the question 6c, briefly describe the mechanisms of end product inhibition. So what is end product inhibition? End product. So when you said end product inhibition, it is actually in the reaction where you have the end product. The end product, let's say you have 5 reactions in one pathway. Let's say you have A convert to produce B, and B convert to produce C, and C to produce D, and D to produce F. So let's say F is the final product. So if F is used to inhibit the activity of the phase enzymes, that is what we describe as end product inhibition. Ladies and gentlemen, it is actually getting late. It is now 10:14, and I think we have done something significant now. We're able to actually solve one question paper. So now, when are we going to meet again? When are we going to answer another question? Our Adebola, I have done, I tried. Why are you saying that I should write on? Okay, tomorrow the same time. Lactate dehydrogenase, Sandra. The isozymes is lactate dehydrogenase. Okay, so no problem. So I will make sure before tomorrow. I will make sure before we have the class. Before we have the class, I will try my best to make sure that I will summarize so many of the video content. At least even if a 1 hour video, even if a 1 hour video, I will try to do the summary of the course. I will try to do the summary of the course. So, but it's only for those that join that will have access to that video. So if you know that you didn't join, please try to join. So ladies and gentlemen, good bless you all. Okay, Onyeka Kenneth, you are welcome. Thank you for joining. So please join your channel. Do we pay monthly? Yeah, it's just 750 naira that you can use to join. So I think it's not expensive because some people are making it even $1,000, $500. Some people are making it $50. But because we are Nigerians, so I just decided to make it just 750 naira so that for everyone to join. Okay, Okoro Onyekachi, I will try my best to see maybe tomorrow 8 p.m. But before that time, I will try to draw a very interesting videos that will also help in summarizing the course as well. And if you joined, there is a video that I actually start solving some of the questions. So please, I advise you after joining to watch that video. I think it's just 6 minute video, and it will be of significant help to all of you. Okay, Abigail Balogun, thank you very much. Thank you for joining, and you are welcome to the YouTube community. You see, the importance of joining the YouTube community is not just only about this course. It's not about this course. It's about even your second semester courses. All the videos will be dropped. All the video on that will be dropped, and you are going to have access to them, and it will really help. How can we locate the 6 minute videos? Yeah, Dorcas, you are already joined. You already joined. You will see that questions in on chemistry of macromolecules. So you are already a member. of the YouTube channel, so you are going to actually have access to it. You will see it after this video. You check on my YouTube channel, you will see it definitely. Okay, so thank you. I think we are going to meet tomorrow. So thank you very much, and I'm going to end the live now. So you can still, if you want to watch it again, you can still have access to the video on my YouTube channel. So please, can you also drop the video on the past question group for those of us that we are unable to join the YouTube community? Actually, even if I drop the YouTube link on the WhatsApp, you can't have access to it because it will not allow you to watch it. It will not allow you to watch it. So that is why I said that you should try to join. You might not necessarily use your ATM. You can use any other ATM to join. So Taibat, you are welcome for joining. Okay, Precious or Serenren. This is the this is my YouTube channel. So once you click on my YouTube channel, you can actually have access to it. So thank you very much, ladies and gentlemen. Good night till we meet tomorrow by 8. By 8. Sandra, try to check. You will definitely join. So thank you. Bye bye. Till we meet tomorrow.